All right. So welcome everyone to today's Big Picture webinar. Today we will discuss SEND and how preclinical data are structured and what will be the impact of that on the Big Picture project. The webinar will be provided by Stefano and Pierre from Roche. Stefano, they're both based in Basel. Stefano is a veterinarian by training and now the SEND coordinator. And he's working with Pierre, who's actually leading the digital regulatory pathology at Roche and is a veterinarian pathologist by training. So some people, there may be some overlap in the presentation for the people who joined the metadata task force meeting just recently. But I think we can use this webinar also to extend on the discussion we had there. We decided that it would be good to have the Q&A afterwards, or you can put them in the chat so they're saved and we can come back to them to the questions later. And I would like to invite you all to turn on cameras so that at least we can see who's there today. I think that's also nice. Just for your knowledge, we do record the session and we can make the slides available after the meeting on the Big Picture SharePoint. So they are available, but not publicly, but only for the people that work on the Big Picture project. So with that, I think I can hand over to Stefano, who will start the presentation.
Yes, thanks. So can you already see my slides? Good, thanks. So the purpose of today's presentation is to give a very basic introduction into SEND. And SEND stands for Standard for Exchange of Non-Clinical Data, and it's a standard which specifies a way to collect and present non-clinical data in a consistent tabulated format using the study data tabulated model, SDTM. SEND is one of the required standards for data submission to the FDA, and currently single dose toxicity, repeat dose toxicity, carcinogenicity, cardiovascular and respiratory studies require SEND. And from March 15th, 2023 on, also embryo-fetal development studies will require SEND packages to be able to successfully submit NDA and the IND applications to the FDA. So who develops SEND? Basically, CDISC develops SEND. CDISC is a global non-profit organizations with hundreds of employees, volunteers, and member organizations around the world. And CDISC is the one who publishes the SEND implementation guides and the corresponding confirmed data endpoints for exchange, the so-called codex. I will explain this in the next slide then. So there are different versions of the SEND implementation guides. Currently, the SEND IG 30, 31, 311, the animal rule, and SEND IG DART are available. And so each version of SEND basically has its own implementation guide. And based on the study type and the study protocol signature date, the corresponding SEND IG has to be followed to issue a correct SEND package. Now the question is, where do I find this information? And this is basically stated in the FDA data standards catalog. So that's a screenshot you can see here. It's not the most recent version issued, but it's good enough to show you what it's stated in there. And basically the data standard catalog publishes when a requirement starts and when a requirement ends. So for example, for the SEND IG version 3.0, you can see that the requirement started on December 17th, 2016, and ended on March 15th, 2020. So basically, send or studies requiring send packages with a study start date within this time frame are supposed to be submitted with a send IG 3.0 version. But there are also kind of some overlaps with 3.1, so potentially both versions could have been submitted. On this slide, you can find some links where you can download the most important documents. So as mentioned before, the FDA Data Standards Catalog, which I just showed, then the different send IGs and the codex. And the codex basically specifies which study design and which data endpoints are intended to be standardized in this corresponding send IG. And that's already a very important point because this basically tells us that not all data which has been collected during your study is supposed to be modeled in send. So for example, currently you will not be able to find immunophenotyping data which maybe has been collected during your study in a send package. This will change in future, but for the currently published send implementation guides, it's not foreseen to be part of the send packages. Then another very important document is the send controlled terminology or the CDISC controlled terminology, which is used for the send dataset generation. So it's a set of CDISC developed or CDISC adopted standard expressions, which is updated and published quarterly. So it's very important to always use the most recent terminology if you issue new send packages, which kind of harmonizes the send data sets between each other. So now what is actually a send package? A send package is a folder which contains different files and file formats. And there are the so-called XPT files in SAS transport file format, which contain the study data in the tabulated format, and these are the so-called domains. Then there is also a non-clinical study data reviewer's guide, the so-called NSDRG in PDF format, containing explanations for the reviewer. So for example, if you have differences between your study report and the corresponding send package, the explanation will be stated in the NSDRG. Then there is also the define file in XML format, which transmits metadata that describes any tabular dataset structure. For example, if a value for a variable has been actually collected or derived from another variable, which controlled terminology has been used, and which format is represented. And on the picture, you can actually see a screenshot how a send package actually looks like with the different domains and the files I just described. So now let's go a step further and take a look at the domains. Domains can be classified in general observation classes and special purpose domains. And the subject level observations collected during the study are represented according to one of the three general observation classes. So we have the interventions, which basically describe, for example, the actual dosing of a drug, or the events which capture the subject disposition, and then very important, the actual findings, which capture observations resulting from tests or evaluations, such as laboratory test results, body weights, and so on. Then, as I mentioned before, there are also the special purpose domains, which contain additional information. For example, we have demographic information in the DM domain, comments, and subject elements, which include study data that doesn't fit in one of the general observation domains. And also a very important point is that there are these trial design model data sets or trial domains consisting of trial elements, trial arms, trial sets, and trial summaries. And this domain really contain everything about your study design. An important point to mention here is that these domains most likely are created manually, so the data or the information cannot be retrieved from the LIMS. And since it's, let's say, manual work, it's a bit, there is a certain potential that errors occur here. Then there are also the related record domain, the supplemental domain, and the pool definition domain. So related record, for example, if you have a certain finding in your macroscopic during your macroscopic evaluation, and you have another finding which relates to your macroscopic finding in your microscopic, so in your histology, then you can describe this relationship basically with the help of the real rec domain. As well as, for example, if you have pooled data from some kind of concentration data, you can then basically describe which samples from which animal are in which pool with help of the pool definition. So here we have a screenshot of the domain overview of the SendIG 3.1. And I personally think that this is a very helpful description. So for those who are new to Send, I would advise always to take a look at the corresponding SendIG, go to the domain overview, and then figure out with the help of this overview where the data you are looking after could potentially be. So it doesn't mean that these domains always are in the Send package. Obviously, it always depends if data has been collected or not. So now that we know what a domain is, we can go again one step further and take a look at the variables. So each observation is described by a series of variables corresponding to a row in a specific domain. So for example, if we are measuring ALT for one animal at a specific point, this will result within one row of your LB domain. And these variables can also be classified into 5 major rows. So we will have identifier variables, identifying the study, the subject, the domain, the pool if there is pooled data, and the sequence number of the record. Then there's also the topic variable, which has to focus on the observation, such as the name of the lab test. In the example I mentioned before, it would then be ALT. Then qualifiers, which illustrate the actual values and the units. For example, for ALT, it would then be international unit per liter. Then focusing on the trial design domains, there are also the rule variables, which are used to describe defined start and end conditions. And last but not least, timing variables, which basically describe the timing of an observation. So now the most important or very important questions is, where do I find the information about the specific variables and the domains? And the answer is always in the corresponding SendIG. An important thing to know is that not all variables described in the SendIG are mandatory. So expected and permissible variables may be or maybe not included in the data set. This always depends a bit on how the study has been set up in the LIMS and also depending on technical limitations. So here, for example, we have a screenshot of our DM domain, and on the top row, we can see our variables. So we have our identifiers with study ID, domain, unique subject ID, subject, and then we have our record qualifiers. So now, if I would like to know what this actually means, I can go to the specific section in the SendIG and basically take a look what, for example, RFST DTC means. And there I figure out that this is the subject reference start day time and in ISO format, and that this is a record qualifier, and this is actually used when to describe when a subject was first exposed to the study treatment. And in the end, we also always see if the variable is required, expected, or permissible. Here again, a screenshot from one of our trial design domains. Here, this is then the trial summary, and the trial summary contains the data which can, the data which can be found in the study protocol. Again, if I would like to figure out what TS-parm-CD means, I can go to the corresponding section in the SendIG, figure out that this is the trial summary parameter shortcode. And if I would like to figure out what the shortcodes are, this can also be found in the SendIG. And with that, I would like to hand over to Pierre, who is going to take a closer look into the MI domain.
Yeah, thank you, Stefano. So also to put this data set into perspective, I think the objective, from the preclinical side is to use the send package to submit some study metadata to the IMI. This is why also the objective of this presentation, because most of the data we need to illustrate, to specify the study metadata are present into this package, like, for example, the species, the age of the animals, etc., etc. So we have a lot of data sets which are per default embedded into this package in different domains. So this is also the challenge at the end, that we need some platform, like for sure the IMI uploader, to be able to retrieve the piece of data we need from each domain to populate accordingly the MS data. I don't recall exactly the name, but the template we have for the preclinical side to give the information which is needed to the IMI to populate the IMI data. So just to recapitulate from the microscopic part, so we have we have one domain for sure, which is called the MI, it's PT. So, and here you can already see from the send document, we have a lot of different variables for sure. Some are present in mostly any domain, like the study ID, but for sure, for the microscopic part, for the pathology part, in this domain, you will be able to get more information regarding to the subject, the tissue, and the findings, because I think this is most important from the microscopic part. So you have all these different variables, as it was described already by Stefano just previously, and some are required. So we will know that we will get this information for sure in the package, and some maybe not. And this is what I also would like to emphasize afterwards, to say that the beauty of the SEN is that we will have an harmonization, let's say, of the data structure to retrieve the data we need, but at the same time, for sure there will be some information which may be present in some SEN package and in others will not, because it was not populated in the same way, or it was not required. So not entered during the study data entry. Next slide, please. So here you see some of the variables which are, you see, it's quite a long list. So for example, which is really important is a MIS-spec. This will tell you which tissue we speak about in the MIXPT with the corresponding findings. And the beauty also of SAN is that this variable, for some of them, are standardized with CDISC, meaning that whatever you have, kidney, adrenal gland, or whatever, the spelling of the name normally will be standardized by default. So it's also something that we should not have to take care afterwards, but the name spelling of the tissue is standardized by default with CDISC, at least before we send this, because we use this packet for the authorities. We check that there is consistency and it is compliant with the CDISC nomenclature. But you have a lot of different additional variables you can have. For example, the specimen condition, like autolyse, may be there, maybe not. It's affected. The laterality may be there, maybe not. It really depends how the data entry was entered in first instance in the pharma company or at the CRO. And for example, you will get the severity for most of the study we have. I'm quite sure of this. So we should be able to get this information on top of the finding if the data is to be provided by the EFPAIM member. Next slide, please. So here you have some concrete examples of how complex the data can be still, because we can standardize some of the variables for sure easily, the tissue name, some specific variable like the distribution or the severity, yes, but the finding itself, you have much more information there. So this is what you see on the top. This is like a little bit like the diagnosis you can have from the clinical side. You have the description of the finding, the initial description into the LIMS system, which was used from the preclinical side. So you have we call the best process, like amyloidosis, grade 2, for example, inflammation. But you may have additional modifiers like inflammation, acute, could be focal, can be multifocal, and this is not standardized by default. The best process can be standardized. It depends on the glossary which was used, but the original diagnosis may have or not have different level of modifiers included. This is why into SAND we have a variable which is called MIS-stress, where you see here on the middle of the table that this, with the latest guidance of 3.1, should be standardized. So we have a CDISC nomenclature where, for sure, for example, amyloidosis or necrosis will be part of the CDISC nomenclature, so you don't need to harmonize. In some instances, this may be harmonized by the sponsor, by us, before submitting the SAN package to the authorities. But if it's a former version of SAN, this may not be the case, meaning that you have a structure, you can retrieve the information from SAN, but that does not mean that the same finding or best will be described the same way at each FPA member level. Next slide, please. So here to show you, yes, how it looks like really more in the XPT file, when we extract the data from the LIMS. You see here you have different columns, so for sure you will have the study ID, you will get the subject ID, and then I think the more interesting for you is the MIORES and the MIS-stress. You see here again the full finding description with MIORES, which comes from the LIMS. This is our laboratory management system where we enter the data, and the MIS-stress where you have we call the best process, and all the other variables either are in this domain, the MI. So, for example, On here you cannot see, but on the left or the right side you will get the severity, for example. And other modifiers are likely to be in over the supplemental domain we call subme. So next slide, please. Here you see the severity on the right, and you see the full and the laterality, for example, which is also displayed or present in this me domain. So sometimes it will be present for that per tissue, for example, but sometimes for sure it may not to be. So this is not sure that you will have the laterality for each of the tissue you have in your study. Next slide, please. And here I want just to show the supplemental domain, microscopic domain, where I just mentioned these additional modifiers, which will be there, with some difference between the same version. For example, in the 3.1, the new guidance, some modifiers like the distribution or the chronicity will have to be populated in the me domain. And this supplemental domain, you will have mainly the locator, the cell type, for example, let's say, or the locator into the tissue. In 3.0, it is different. You will have likely, if present, the locator, the chronicity, the distribution into this supplemental domain, which means, in terms of data extraction, that we should be able to adapt our extractor, the IMI uploader, depending on the send package, because I really think that the FPA members will use send data package to submit data to the IMI because it's much more simple. It's much more harmonized than the former data set we have. But we need some flexibility here. And so this likely to be at the AFPA member level when we define this IMI uploader, how we want to extract the data from which field, from which variable, and depending likely also the send version we have. Next slide, please. Yes, I think so. This is the end of my presentation. So at the same time, it's quite simple and complex, I would say, because simple because we have the opportunity from the preclinical side to use the send package, which adds a lot of let's say uniformity in terms of data structure. But at the same time, when you retrieve the data set, for sure, you still have some significant difference in terms of data population, at least from the pathology side. From the, let's say, more critical, basic domain, perhaps Stefan can precise this, like the study type, likely it's more easy. I mean, it's more can be more standardized from the pathology side. But for the pathology, for sure, we will have some heterogeneity of the data to be extracted. And I think this is all from my side. And also, this is why we are working quite extensively with FPA members and Catriona from Deciphex to define which variable, which fields are critical for IMI, which are the ones which are mandatory, and which are the ones which are optional. So, for example, for sure, the tissue, the type of staining you have, there is clearly standard mandatory field that we want to populate for the IMI data submission. Some others are likely to be only optional, but this is being discussed intensively also within the team, the preclinical data team into IMI. And I will be curious to know exactly how you would like to proceed from the clinical side, because this may be different also at each, I don't know, university or level, or from the at the laboratory level, how different are the data set and how you can extract and submit them to IMI.
All right. Thanks a lot, Pierre. Thanks a lot, Stefano. So the floor is open, of course, for questions. Please, you could raise your hand, but also just unmute and speak up. I think that makes it most easy and most interactive. So curious, are there any more kind of basic questions people have on scent? Oh, very clear. That's good.
Very clear or very confused one. So,
The other people may be able to answer that question. So I did have a question, and that is, it mostly relates. Of course, we want to build a repository with data that's valuable, that can be used for AI development, and to build tools that well will make our work easier and better. From your perspective, what are the data elements that are not in, or would you say that all the data elements that we need for AI development are in Cent, or are there specific elements that you think this is still missing, but that well may or may not be implemented in the future? What's your take on that?
I think from the pathology side, I mean, the basic for sure are there. The tissue, you may also have the findings or the description of the finding. Probably we need to specify the staining because it's not sure that this is always into the Cent package, even if I expect really most of the slides to be standard HME slides. There is another topic which was not addressed here is the scan file metadata itself. Because for AI, for sure, you need some basic metadata information to be there, still there, like the resolution, etc., etc. But it's a separate topic. I think for sure from our side, the description of the slide, I guess, if the sponsor is willing to share, I mean, I'm sure that the information you need will be there. From a technical side, as a it's more about the scan file that metadata we still need, because one important topic we did not see discuss here is I know we have new terms to come for the anonymization and pseudonymization for the technical data, but for sure there will be anonymization or pseudonymization of the data. For IP matters for sure, this is clear. And also, yeah, I mean, for AI purpose, if you know the tissue, I think the big challenge we have, and this is where we need to be really diligent from the preclinical side, is we now to be able to make the match between the scan file and the tissue, which is not so easy per default. You need to get a tool, a IMI uploader from our side, which allows to make the link between the scan you see and the tissue you have. And you may, sometimes we have multiple slides for one tissue. This may be the case. Or we may have also different tissues on the same slide. So this is really this metadata match that we need to take care. And I'm confident that when you have a process in place, especially with label barcodes in the lab, then it's much easier to get this correspondence. So this is also an important factor for us to not to go into manual steps, or to get as less manual steps as we can to get a reliable process in terms of data population and also this data match. So hopefully we are already using Send for using visualization tools from the preclinical side, where we already have this data match, meaning we are able to select some slides for one specific animal number and one tissue. So we should be able to do the same for IMI, meaning that you want to get a special strain and special tissue, then the IMI visualization tool should be able to do it, because we have the correct metadata structure and gathering before submitting the data to IMI. But I think if this is really for the metadata part of the scan file, I'm not the expert, but I'm confident that we'll get enough information, and at the study level as well, because we have all the study data which we need from the regulatory side to be submitted. So there is no reason that we could not get this data. Like, for example, very important, I guess, would be the strain, the species, for sure, the age of the animal, so we can give a range. And afterwards, it's quite simple. The tissue, the staining. For classical studies, this is okay. If some sponsor want to submit special stains or immunohistochemistry, this is different. I guess here it's not a standard study, SEN study, let's say. So then you probably have to go into a manual stage there. And for sure, this will not be into the SEN from my perspective. But this is another topic.
Maybe to add on that, what I think will also be quite challenging is that you will get data sets from different sponsors, and there will be a certain heterogeneity. So basically also all variables which are kind of permissible. So from one sponsor, maybe it will be included, from another sponsor, maybe not. And another thing, also the different versions of SEND. So basically also with the updated control terminology and so on, it will be quite tough to, let's say, evaluate data from different sponsors across the industry. So this could potentially be tough.
I see. So one follow-up question on that aspect, Stefano. So when
excuse me, Rick, I just wanted to react to Stefano's just last sentence. So this is why I think it's very important to correctly assess the mandatory metadata that we are preparing. So it's really at this point that we can really drive what we'll be able to do with the data after. Sorry, Rick. And there is Moritz also who wants to intervene.
Yeah.
Yeah, it's not very important. Just and you touched on this already, but there is a one aspect, one common and important known problem with the modifiers for the microscopic findings, which is that the some findings, so the findings that are codified in there are on a very high level, and that's very good from a data scientist point of view, but some modifiers are extremely important in some cases. For example, the common example is inflammation, where the controlled term in SEND is actually just inflammation as is, without any codified modifier, and all the modifiers have to go into this bucket called Mirus mod, which is just all the rest, that's so this is a non-controlled terminology field, so wild west, and everybody's probably going to be using very similar terms, but it's not completely sure. And for most or for many findings, that doesn't matter much, but for some findings like inflammation, this will open up a wide variety, or maybe not that wide, but a variety of different terms that could be entered and cause the usual problems with search and so on. And yeah, just to add on to Stefano's variety point.
I think the Mirus mode also, it's likely to be the case for 3.0, but for 3.1, you have, for the chronicity, for example, distribution, it will be specific variable. There are specific variables into mean. So also that the data population may be different. And I think probably we need to be pragmatic there. we cannot, let's say, guess what be the level of granularity you will have from each study, preclinical study, for the inflammation description, for example. Some may be very descriptive, some a little bit less, and this is something that we need to take account for the development of algorithm or whatever. This is clear for me. Like you mentioned, I mean, this is there may be heterogeneity there. Yeah, and I think you also some question in the chat from Roof, or remark from Roof and Fauve as well. Yes, Wolf saying that the issue of multiple slides per tissue lacking information regarding which slide exhibits the lesion seems to be a potential challenge. Absolutely. I can give an example. If you have the brain with 4 different slides and you have gliosis, which is described, I don't know, gliosis focal, it's likely to be, you will not know on which slide the finding is. Because by default, we do not specify in our data recording. It could be, but it's likely to be not the case. And then, meaning that if you want to run an algorithm for gliosis, you need to annotate, but probably you have to look through the 4 or 5 slides to see which one the findings is referring to. I think Catriona has a question.
No, it's just a comment in response to that question. So being privy to different conversations, it's a known issue across all of the nodes, including clinical, that the finding won't be on a slide level. It'll be more of a summary finding. So say if you're an end user of the repository and you search for a particular lesion or disease state, you'll be presented with images that relates to it, but not all images will have the lesion or finding that you were looking for. So you'll know it's contained within some of the images being presented, either by a flag. It's an issue, I believe, that clinical are currently evaluating also in terms of how to mitigate, but it's consistent between clinical and non-clinical that the findings or observations won't be at a slide level currently.
Which is good in some ways. That it's the same level of description at the slide level, let's say, or not between non-clinical and clinical.
Yeah.
Hey,
So
maybe one quick
yes,
question.
Yes.
Yeah, hi. And so, and I'm sorry if it was mentioned and I missed it, but is there like a default implementation of a parser or something to parse the entire nomenclature of the SAN standard? Or like basically, are there machine tools currently available, open machine tools to parse all of that?
I'm not sure I got the question. Because I mean, from the practical side, when we populate this finding at this stage, really the recommendation for sure is to use the SAND or SEDIS nomenclature. So, but afterwards, I don't see how the parsing could be done. I'm not really sure to see what we could do there.
But basically, the parsing is done while there is the SAND export of the study data.
Yes, you're right. So, meaning when we create the SAND package, the initial diagnosis, if it's not named or SEDIS compliant, should be mapped to a SEDIS term. This is what you mean, Ariel?
Yeah.
Yeah. So yes, you're right. This is how it should be done, and we have what we call some SAN shaker, like Pinnacle 21, where we can, at least with 3.1, we can check the compliance of the finding term in the SAN package, meaning that does that fit with the SEDIS nomenclature, which is evolving. Every trimester, we have new terms which are populated, which are implemented. So this is a lively nomenclature. But yes, but for 3.0, this is not likely to be the case. We did not have to make this mapping per default for microscopic findings. So that's why also there will be a difference between 3.0 and 3.1.
Okay, thanks.
But overall, I think it's quite from some years that the in-end nomenclature is used either by the pharma companies or the sponsor or the CRO. So this mapping generally, I hope, is not so, let's say, intense before getting the SAN package, because normally in most of the cases, the in-end nomenclature was used by default.
Normally it generates errors if the mapping is not good. So then generally the internal submission groups don't like them, and they go back to the pathologist to say, hey, you have to find a solution. And little by little there are less of those errors. At least this is what we experienced here.
Yes, the same from our side. Yes. And I'm curious to know how it works from the clinical side. I think these findings, how is it? I think this is standardized for sure, but I'm not the expert here.
There's somebody here who can take that question. Maybe Joël or Raphael.
I mean, it does not matter if we can have some glimpse of information after the webinar. It's perfect. Just for curiosity.
Yeah.
So we've put forward a template to consolidate all that information. I don't believe anyone has worked on it just yet, but it is up on SharePoint where we have effectively those questions for each of the node asked. So it's in the metadata task force version 011, I think it's EOG specification, if people want to have a look. So we've requested that each node fill in columns in terms of the nomenclature, ontology, vocabulary, if there's standard format, standard vocab, and whether or not fields require anonymization, or if they can be open. So that might answer some of those questions. Just a high-level answer to the clinical question, and again, this is just from being present on some of those calls. My understanding is there's no standard per se for findings or observations, but again, it would be more of a summary finding in terms of a case rather than findings or observations on a slide level, if that's the question. Thanks, Wolf. So Wolf has popped the link into the chat for the MSM DAD template that I was talking about just now.
Yes. All right. Are there any questions, comments left? Anyone? No. Okay, then I would like to thank Pierre and Stefano for taking the time to explain us about all about scent. So I think it was very helpful, and we'll hear a lot more about it, I guess.
I prefer it's them than me, frankly.
It's a shared effort for all of you members anyway. So, but that's exciting, but also challenging at the same time. Curious to see at the end how the process will work, because it's very also interesting brainstorming effort from our side as well.
Yeah, and I think this challenge it's there on the non-clinical, but it may be even bigger on for the clinical data. Okay, so just as to wrap up, we have 2 more webinars coming up before the summer break. During the summer, we will, I think, skip 1 or 2, but I will let you know by email. So the first one in May, we're not entirely sure, but it likely be Julie Boisclair and Brian Knight, who will talk about and discuss with you everything you always wanted to know about toxicology pathology, but never dared to ask. And I think this is in particular also very interesting for the people not working in toxicology pathology, just to create more common understanding on what is it actually that half of the project, half of the people involved in this project are doing on a day-to-day basis. So I think it can be very insightful, and we hope it to be very interactive too. And the second one is scheduled for 10th of June. And that relates to a deliverable we have submitted to IMI already. That was prepared by Work Package 5 by Gabriele and Xavier from also from FPI companies on the landscape of guidelines, standard and regulatory requirements relevant for digital pathology in a non-clinical setting. So if you want to join the webinars, please register using these links. They should also be in your email box, or shoot me an email if you don't have it. One more slide, and this is we would like to make these webinars about every month, and it's up to you also to come up with proposals for new topics. So either it's something you want to know or something you want to share with the rest of the consortium. So please shoot us an email if you have any suggestions, and we can see how we can fit it into the schedule. I think with that, I don't know if there was something in the chat still. Yeah, exactly. There's a link to the domain on SharePoint where all the slides will end up, as well as the recordings to look them back in case you want to do that. Thanks a lot again, Pierre, Stefano, and all the others for joining and for the interesting discussion. And I hope to see you next time.
Thank you very much.
Have a good day.
Bye-bye.
Bye.
Bye.
Thank
Thank you
you.
very much. Bye bye.